The other thing is I'm recording all these lectures, and we'll get to that as we move along here. I'm also going to try and run it from my phone, which may or may not work. We shall see. Can everybody see the slides, actually? Is that good enough in the back? Yes? Good? Okay. So who am I? Those of you are fortunate enough not to know me. I know a few of you already. I'm a professor of biology here in Portland State, been here since 2001. I know that for a fact because I moved into my house on 9/11. So I know exactly how long I've been here. Where can you find me? By far and away, the easiest way to reach me is email. I'm pretty email dependent. So kstedman@pdx.edu. That's, again, by far and away the best way to reach me. Any questions, comments, worries, etc. I'll try not to answer them during class, but other than that, good timing. You can look for me over in SRTC, room 466. That's where office hours will be immediately after class, or just right here. I don't know if there's another class after this in here. If there's not, it's great. We can just stay here. If there is another class, then we can just go over there. You can see things that I post on Facebook if you like. Actually, my lab set this page up for me. It's mostly stuff about viruses because that's what I do. I'm kind of what people call a virus hunter, among other things. That's the real activity of the research lab. This actually is a poster some of you may see on campus every once in a while. Hang on. Yep. See, this is the problem with trying to run it from my phone. There should be a pointer here. It's not coming up. Oh, well. Also recently got dragged kicking and screaming onto Twitter. I figured if the president can do it, so can I. And yeah, I try not to be political in this class. I don't always succeed. But so I'm on Twitter. In fact, I just posted a picture of this classroom on Twitter. So, but you shouldn't be checking it during class. Mostly, again, I post stuff about viruses, fun kinds of things in molecular biology, stuff that I think is interesting from a professional point of view, and again, I try not to be political in that particular case. YouTube, I have a YouTube channel that will probably be important for a lot of you because that's where all of these lectures end up. You can feel free to subscribe if you like, that way you'll get an immediate update on whenever I post any of those lectures. My website is up here as well. Unfortunately, it's ridiculously out of date. I have somebody who's trying to update it for me. Oh, there's my pointer. Here it is, K. Stedman. Again, eventually that'll happen. If you want to talk to my voicemail, call my phone. I almost never answer it. So you can try, but that's unlikely to happen. I also, full disclosure here, I started a company about 3 years ago on vaccine stabilization. Vaccines are amazing. Hopefully all of you have your flu shots this year. Yes, it's not the greatest match, but it's still a heck of a lot better than no protection. And that's not a political statement, that's a scientific statement. And then for a while, I've been working on this documentary movie on how amazing viruses are and how they really have kind of a bad rap, but that's unfortunately not as up to date as I would like it to be as well. A couple things about the course. This is really a fundamentals of molecular biology course. I like to think of this as the basis that you can build other classes on, and particularly the only other class that makes any sense whatsoever is virology, which I'll be teaching next term. So a lot of the basis really of molecular biology we'll talk about here, and then try and transfer that hopefully to whatever more advanced courses you're doing. So we'll spend a little time talking about chemistry and genetics. That should all be review, hopefully, because all of you have taken principles or the equivalent thereof. Then we'll spend quite a while talking about DNA and RNA structures. I have my model, which I forgot to bring today, but I'll try and have on Wednesday and then all the way through the first part of the course. It's actually got some really interesting problems with it, and so I'll bring and have people have a chance to take a look at that as well. I'll also talk a little bit about genomes. Genomics is a whole course in and of itself, and you'll probably hear me say this multiple times over the next 10 weeks, is this is something we could spend 10 weeks talking about, and genomics is definitely one of those subjects. So that'll sort of be the beginning part of the course. Then we'll talk a little bit about how to make more DNA, so DNA replication. And one of the really wonderful things about DNA is it's really easy to make copies of, and also it's not a great molecule for genetic information. It just so happens that that's what we ended up with. And so we'll talk about the repair process, which is a problem as far as DNA mutations mostly being bad, but is also really important from the point of view of evolution, because if we didn't have any kinds of change, evolution would basically not be possible. That point will have the most interesting part of the class. No, one of the three most interesting parts, that'll be our first midterm after, again, DNA, RNA structure, and a little bit of replication. Then we'll talk about transcription and translation. This is all going from now DNA to RNA, and then from RNA on to protein. Again, RNA here is one of those subjects that we could spend 10 weeks talking about. And there are some really nice classes in chemistry, interestingly enough, that talk about RNA and RNA processing. Then we'll have our next midterm. Then we'll talk about regulation, and this is how genes get turned on and turned off. Yet again, how many weeks have we spent talking about this? 10 at least. And then a little bit about RNAs, and particularly small non-coding RNAs, but also large non-coding RNAs. These may or may not mean something to you at this point, but it turns out that RNA is way more interesting and important than we ever thought that it was. So that's more or less the overview, what we're going to cover in terms of topics. In terms of what I expect you to know, there are a couple of people who've asked me about this online as well beforehand. We're not going to get any beyond general chemistry. So hopefully you've all had general chemistry. If you haven't, the first part of this course, and actually later on, will be definitely a challenge. And if you haven't, please talk to me, send me an email about it. Principles of biology. This is biology 211. I know some people are taking 212 right now. That's not great, but it's okay. It's certainly doable. A lot of this stuff will be building on what was done in 211. Helpful but not required. How many of you have taken or are taking genetics now? Okay, so there will be a considerable amount of overlap between the two, hopefully not too much. And if I disagree with your instructor there, I'm always right? No. Please come and talk to me about it, because there may be some differences of opinion, and I quite possibly could be wrong about some of these things. OChem, how many people are taking OChem? Okay, potentially useful, but we're going to talk about like 3 different reactions in this class, so not very many. Biochemistry? Okay, lots of overlap between biochemistry and here. And now, don't tell any of the chemists, but if you've taken this class, biochemistry would be really, really easy. Shh, don't tell any of them. Then microbiology, not so much. Anyone taking micro this term? Way down in CLSB. I'll be doing a guest lecture down there on viruses at some point. Should be kind of fun. Cell biology. Few of you. I know that Dr. Singer says that you should have molecular biology before you take cell biology. That's partly because we both use the same book. And the first part of the book is molecular biology, and the second part is cell biology. But if you have any questions about these prerequisites, please send me an email, talk to me a little bit later on. So what do we use for the class? I suppose I could turn this on here. Hopefully all of you have this book, Molecular Biology of the Cell. It's coming up over here. 6th edition. A number of people asked me about the 5th edition. I'm going to try and have a copy of this text over in the library. Has anyone looked for one over there? Okay, there may be one on, sorry? There is one on there. Okay, I was going to go and check again. See if we can have a few more. There are, I think, 170 people registered for this class, so I think we should probably have a few more than that. And it's on 2-hour review for that. Couple of things about that book, and I guess I mentioned, some people asked me about the 5th edition of the textbook. Molecular biology is amazing. It moves so incredibly fast that this textbook is actually really out of date already, even though it's from 2014, and particularly a lot of that stuff about the RNA that I was talking about. And what that means is the 5th edition of the textbook, which is from 2008, is even more out of date. There's a lot of things that are similar, and I'll use some of the images from that textbook as well, but this is, and they're working on a new version of this one as well. It's every probably 4 to 5 years that this whole thing turns over. And I'm going to try and incorporate quite a lot of that newer stuff into my lectures. But that's the basis here. What are we going to cover from the textbook? It's basically going to be chapters 1 through 8, and really skimming over 2 and 3, because a lot of this should be background for you. There's a course website. Everyone gone on to D2L? Yes, found stuff there. There's a bunch of additional materials there. The main important things are the PowerPoints. So everything that I'm showing here will be posted on D2L, hopefully by 10 o'clock the night before. I make no guarantees on that, but I will certainly try to do so. Syllabus is there, so if you need to go back and check, look at things on there. A few notes, but probably most importantly for a lot of people here, all of my old exams are there. And people always ask me, what's the best way to study for Stedman's exams? Go back and look at old exams. So the old exams are there, all the keys are there. Sometimes I forget to post the most recent exams without keys. So if you're looking for something you can't find it, again, send me an email, let me know. And again, all the lectures, which, and I'll back up a little bit, those of you who haven't had me before, which are probably quite a few of you, what I do is I'm recording my voice and all of the slides up here. And so that's what I post on YouTube after class, usually by the afternoon after. I've found that people really like to go back and listen to the lecture again. Why you want to listen to me more than once, I don't know. My kids certainly don't, but it seems to be really quite worthwhile, and at least at 1.5 times speed or 2 times speed gets you through it relatively quickly. So that should all be there as well. Molecular biology, as I mentioned, is a very, very fast-moving subject. There are older textbooks which are out there. Lodish is a 2012 version. I think there may be a newer version of the Lodish textbook which is out and available. Certainly, a lot of the stuff that we cover here is going to be covered elsewhere as well. Yes, the 4th edition and 5th edition. Interestingly enough, I think the 4th edition of the textbook is completely freely available online at NCBI. So if you want to get some of that very background stuff, again from 10 years ago, but some of the background basal stuff there hasn't changed that much, and that's completely available. So that's right here. But again, lots and lots of other textbooks, some of which you can find in a library, some of which I actually have copies of in my office and can loan you for short periods of time if you're interested in that. So what do most people care about? How do we do evaluation? Necessary evil, unfortunately. The way that I do my exams, there are 3 of them. They're not cumulative per se, but I assume that you know the material from the first one on the second one and from the first and the second on the third. The way I do that is 2 midterms and a final, each of them 50 multiple choice questions. So the reason that it's 50 multiple choice questions, I hate multiple choice question exams. I hate writing them. I hate taking them. But I have 170 people in here and no graders. So if you want to actually get a grade before next term, I kind of have to do the multiple choice thing. So that's the way it is. And Scantron's the big 1-page version of those Scantrons. Clickers. How many of you don't have clickers? Okay, you will need them for this class. And I apologize for that, but it's actually really useful for me as well as for you, I think. One of the things that is a little different in terms of clickers, I want you to talk to each other when you have clicker questions. So please, sort of an idea of, find somebody who's getting really good answers at clicker questions and talk to me. Yes, question?
Can we use the app?
You can use the app. Yeah, unfortunately, a couple of us in biology have talked about this. No, you can't use the app. And the couple of reasons for that is one, yes, you'd have your computer, which is right there with the app right on it, or your phone, et cetera. And so that's one of the reasons. If anyone actually, by the way, thank you for asking that question, has some financial issues with actually getting a clicker, let me know and I may be able to help you out. Again, send me an email about that. Okay, and just to force you to do clicker questions. Usually clicker questions don't end up changing anybody's letter grade at the end of the term, but you do, it's just going to be 10% of your grade. But again, find people who are smart and sit next to them. So, that being said, we'll have our first clicker question. But this one does not count. But it's basically, it's a way that we can see that your clickers are actually working. So I will post these all online. It's the AA frequency, by the way, here. So are we all ready? Yes? Clicker questions. Which of the following best describes you? And I know some of these may be not so good. Junior biology major, senior biology major, post-bac, junior, senior, non-biology major, or Stedman's favorite answer, which is, hang on, base frequency, yes. It hasn't actually started yet. Sorry about that. Where are we? Clicker question. Where's my clicker thing? Where'd it go? Oh, it just stopped. Okay. One instance of iClicker running. See, that's what happens when I start with a new classroom here. Do you guys see a clicker? Your clicker's registering? I'm not getting anything here. This is very strange. There's always something about this. Okay, new session. Try again. That looks better, doesn't it? Or not, as the case may be. See, this is why I do this before they actually count. Try. Is it going to start? No, we're still not starting. Okay, let's just start this. Still not working. Hold your power button for 2 seconds. AA, set. Still not going? There we go. Yay! Hallelujah. And the way I'm going to do clicker questions is also just a countdown. And if we get to a certain point with clicker questions that people are like, I need more time, I will give you more time. So this also gives me a chance to take a picture of all of you. If it'll go. You'll all be on Twitter. You'll all be on my Twitter account. Yes?
I tried to register clicker on the iClicker website last night, but your class didn't pop up. Is there a better way to do it?
So yeah, registration. In fact, that's basically on my next slide here. That should, at least in theory, be working, but I may need to synchronize with D2L first. So if you had trouble, anyone able to register yet? Not yet? Okay. I will try and make sure that that actually ends up happening. But yeah, I may need to update the class to D2L, which is another reason to ask these questions here. But let's see what you people think. Okay, so in terms of who is whom, because you guys are probably interested in this too, and I will always show the answers to questions. I won't show the correct answer to this question, at least not here. So about 40% juniors, 25%, if I can see the numbers here, seniors, 15% post-bacs, 18% non-biology majors. I'll find you all. And then none of the above here. So that's really good for me to know. And one of the things that's happened over the last few years is we had more and more juniors taking this class, which is great. We used to have a ton of seniors, which was really bad because they should have had this course before they were taking other courses. So that's the process here. So now we should close these, get rid of the results. Okay, so yeah, so register them, and I will check and make sure that that's working. And if it is not working after today, please email me and let me know, because I will then try and take care of that. And I'm sorry about the registration fee if you have got to use Clicker. That's completely beyond my control. So, but yes, let me know if we're having trouble registering on the website after this afternoon, because I'll try and upload these points. So the other thing with this is I will upload, so you know that your clicker is working. I'll just upload these scores today. And so each of you who's here actually clicked will get that. Grading. I've had a couple of different grading schemes over the years. This is the one that seems to work the best and has the least discussion about it. Any of you who've talked to people who've taken my classes before know that my exams are, what are they called? Ridiculous, horrible. There are other kinds of things as well. But what that means is the high score is usually about a 40 out of 50, which means I've written 10 bad questions. But what I do is then I normalize that. That counts as 100%, whatever that high score is. And then I scale from that. And I will tell you what those high scores are, and so you will know exactly where you stand. And I do exactly the same thing with the clicker questions as well. So that's, that should be pretty straightforward. Again, all this stuff is in the syllabus. How do you do well on those exams? I mentioned this already, but one of the really main things is go and look at those old exams, because I try not to reuse questions. But I've gone back and looked at some of the old exams and my new questions, like, wait a minute, that's like exactly the same question I asked before, or very, very similar. People have said this, again, I don't like to do this, I hate to hear my own voice, but go and check out those lectures on YouTube. That seems to work really well for people. Office hours, again, after class, that'll be either here if there's no other class, or over in my office afterwards. How many people have a class right after this? Okay, so there are quite a few of you. So we can definitely also make appointments and have outside of that regular office hour time as well. So that is certainly not a problem. I highly recommend study groups. There are a couple of apps, the Buddy Up app, I think, that helps with study groups. Otherwise, D2L is a great place to post and say, I'd like to try and get together for a study group. There's no better way to learn this material than to try and teach somebody else it. And that is by far and away the best way to do that. And often what happens is I'll have study groups then come and talk about things first and then come to my office hours and go, Dr. Stedman, you explained that really badly. And they're usually right. And so I try and do a better job later on. Discussion tool on D2L, one of the things that happens, as I say, I'm email dependent, so people will email me questions. I'll answer those questions, and I'll post the question and the answer on D2L. There's a questions section in D2L in the discussions point. So I'll anonymize the question and then just put up my answer there, and some people find that really helps too. I also do in-class reviews. You may have noticed if you've looked at the syllabus already, the last lecture before an exam is always review. And that also gives me a chance to go back and talk about some stuff that I think I did a crummy job of explaining the first time around. And some of you have already got it. The best way to slow me down is to raise your hand and ask questions in class too. If you have a question, it's highly unlikely that 169 other people don't have a similar question. Okay, so I have to go through this. If anyone's cheating on an exam, they get a 0. You can do the math. That'll tell you where your grade ends up. I don't insist on people coming to class. Clickers, yes, 10%. You can miss 10% of your points and still get an A in the class. It is 90% and above is an A of whatever the highest score is. So it's definitely doable. But I did collect some data. Like all scientists, I'm a total data junkie. This is from a few years ago, and actually before I had clickers and before people got points for actually answering clicker questions. People who almost always came to class, this blue thing here is the people who got A's, red are B's, white are C's, and anything below that. So almost always, those are when most of the people got A's. Doesn't guarantee that you'll get an A by any stretch of the imagination. People came to class all the time, so got a C. But you can see that that proportion goes smaller and smaller if you go most of the time, sometimes, and rarely. So, but you can still get an A and rarely come to class. It's up to you. Now, you're paying your tuition or somebody's paying your tuition. That's completely your call. Makeup exams, I do do make up exams if they're really extreme circumstances, and I'm going to need extreme circumstances. The main reason for that is it's fair to everybody else. Everyone else is making an effort to be here. And so if you feel that you don't want to take the exam on a particular day, that's not a really good excuse. And if we have this kind of thing, I will make a make-up exam. It's going to be an essay exam because the multiple choice exams everybody gets to take with them. So it's a very different exam, and ask people who've taken them before how they like them or not, as the case may be. Syllabus, again, I was on the website. Go to D2L. I was thinking about going to D2L, but figure we don't really need to do that here. Everyone's pretty clear on D2L, hopefully. Ask me questions. The clicker is as much for me as it is for you. This way I get an idea if the majority of people are understanding things the same way that I'm understanding them or how I've discussed them very well. The discussion, this is with the discussion group on D2L. Again, I'll be posting all of the questions, etc. there, and the way to reach me is email. Some of you may have noticed, at least in past years, I've had a pre-quiz. This is a really easy way to get points. So this is to give me an indication, a little bit like the clickers, on where you're at in terms of the material and the material we're going to cover for the whole term. This is my pre-quiz, as it were. It's 10 multiple choice questions. They're going to be really similar to multiple choice questions that end up on exams. And I don't expect you to know the answers to most of them. If you do know the answers to most of them, they're like, oh, Stedman, this is ridiculously easy, then maybe we should talk about you not actually even being in this class. But if they're really challenging, great. That's exactly what I'm looking for, because hopefully that's some of the material that we will have covered by the end of the course. Anybody, once you start, you have 10 minutes to finish it, 1 minute per question. You can go and look in the book if you want to figure out all these things. That's not very useful because I'm going to say, oh, this person's really good at looking something up in 1 minute. So just go through it, give me an indication of what things look like. These will not be graded. It's just if you start taking the quiz, you'll get 5 points. You can just start the quiz and then stop the quiz. You'll still get 5 points for it. And again, I expected these questions. These should be challenging questions. And they should be something you're like, that sounds interesting. I hope Stedman was going to talk about this. That's the plan anyway. Okay, any questions about how the course is going to, or at least hopefully, run? D2L, yes, sure, thanks.
I have a question about the quizzes. Is it a
Yeah.
quiz that'll open after every class, or is it once a week?
So this particular pre-quiz is just now. So it's going to be now through next week, and that's it. That's the only quiz that we'll have for the term.
Oh, okay, so
It's just one. And all it is just, it's basically to give me an indication of where people are. So for instance, it's just like one question that everybody gets right. Like, forget covering that. We'll cover some more interesting new stuff. So it's really for feedback for me. Other questions, comments, worries, concerns, etc. Again, email is by far and away the best way to reach me for this. And I may walk up and down here. At least this way I can see people. If I go down there, I can't really see people, which you may or may not think is a good thing. Okay, so I'll start my lectures, the normal lectures, instead of talking about class, with an outline of what I'd like to cover in that particular day, and depending on how many questions, we'll see how it goes. You'll notice looking at the syllabus that things can get out of whack pretty easily here. First, talk about the 3 domains. People know about the 3 domains? Yes? Good. 3 domains? Okay, so very often through the course, one of the big deals with molecular biology is it's the quote-unquote same for everything. That's not completely true, and we'll spend a lot of time talking about some of those differences. But archaea, bacteria, and eukarya. I work on archaeal viruses, so archaea are very near and dear to me. A lot less is known about archaea than about bacteria and eukarya. But a lot of these sort of overarching themes are things that we're going to be talking about again and again throughout the rest of the course. Central dogma, hopefully, is something that everybody knows about. DNA goes to RNA, it goes to protein. Works most of the time. And then we'll talk a little bit about DNA, RNA, and protein. Again, most of this should be review. And if we get a chance at the end, we'll talk a little bit about genomics. So you'll also notice that sometimes my lectures are a little out of sync relative to the text. This is what I think makes more sense from a lecturing point of view. This is a particularly good example of that. They bring up the tree of life much later in chapter 1. I think it's much more fundamental, and so we should talk about it right at the beginning. So how many of you have seen trees that kind of look like this? Yes? Good. So this is a molecular representation on how sequences are related to each other. When I say sequences, I'm talking about protein sequences, nucleic acid sequences. And if two sequences are more similar to each other and have come from particular organisms, you can assume that those organisms are actually more similar to each other. And there are problems with this kind of analysis as well. But basically, if two sequences that have come from two different organisms are similar to each other, those two organisms are also closely related to each other. And what you can do is just look at lots of sequences from lots of different organisms. They'll have certain distances, changes in sequence between them, and you can put them into a tree because you can then assume that one organism gave rise to a different organism which gives rise to other organisms. This is all from Darwin. I'm working on the Darwin beard here. That's my plan for the rest of the term. So Darwin came up with this idea of the tree-like distribution of life. And if you look at all life down here at the bottom, there are really three main divisions: eukaryotes, archaea, and bacteria. There are lots of gray zones in here because we're not really quite sure what's going on. But also, it's clear that all, at least cellular life, came from one particular kind of organism. And this is what people call LUCA, the last universal cellular ancestor. In fact, one of my buddies, Patrick Forterre, came up with the term originally. But the reason that we know this is that all cellular organisms are using the same kind of genetic information. They're using the same kind of translational processes. All of these things are really similar. And the most parsimonious argument, or explanation I should say for that, is that they all arose from one particular organism or way of doing things at a molecular biology point of view. And there are other explanations for this too, but most of those aren't very scientific. This is the tree that they present in the textbook, coming from this single ancestor right here. This is incredibly controversial, exactly where this single ancestor was, related to bacteria, archaea, eukarya. Here we have a branch of the eukarya coming out of the archaea. This is also highly controversial, and we can talk a lot more about that as well. Did want to mention what I like in terms of this tree here. This is a tree that was published by Norman Pace, actually almost 20 years ago now. So can you guys see it in the back? Works? Okay, so basically this is what's called a non-rooted tree. It's just the relationship of all these different organisms and not making an assumption in terms of where that last common ancestor was. And I think this is a better way of doing these kinds of trees and descriptions, because you're not assuming that there was a root in one individual common ancestor. But you can also see here that there's a bunch of sequences that are similar. Again, the lengths of the branches between these things are the differences in the sequences. You got one group of bacteria, one group of archaea, and one group of eukaryotes. And most important here, that I wanted to point out, and they talk about this in the textbook as well, you can see it here quite nicely, or also down here, is that what we think about in terms of interesting organisms are molecularly pretty darn boring. At a molecular point of view, we're basically identical to plants and fungi. At a molecular point of view, there's way more diversity, and I would argue interest, in the bacteria and the eukaryotes. Bacteria and archaea. What am I saying? Bacteria and archaea, go back and edit that out of the lecture notes. So much more molecular diversity in archaea and bacteria than there are in particularly what people call the crown groups of eukaryotes, which are all the animals, all the plants, and all the fungi. And those are represented here. When it says right here, humans, that's actually all animals that are in that group. Here, yeast, all fungi. Maize, all plants. So gives you a bit of an idea what's there. But be that as it may, we're still using a lot of the same stuff. DNA, RNA, proteins, etc. So we'll spend a lot of time talking about those individual things as well. So I mentioned here in that previous slide conservation. What do I mean by conservation? This is getting back to this whole idea of sequences. So if you look at particular sequences that have fewer changes, say between particularly different animals, mice and humans, just as an example, molecularly those are very similar to each other, so the sequences are going to be very similar. On the other hand, if you're talking about something like humans and my second favorite organism, Sulfolobus sulfatericus, which comes from volcanic hot springs, these are going to be much more different from each other in terms of the sequence. And in fact, in some cases, so different that you can't even look at how similar they are relative to each other. So what you need to do to make those kinds of trees that we were talking about on, well, I was talking about maybe, on that last slide, you need to find genes that are similar for the kinds of organisms that you want to compare to each other. As I mentioned before, we've got DNA, we've got RNA, but what molecules are the same in all of these cellular organisms? Ribosomes, processing. So the ribosome is conserved, and probably there's a ribosome way back in that LUCA, the very bottom of that tree. So how you look at something that's going to be comparing organisms that are very distantly related to each other, you need to find something that's conserved, i.e. is present in each of those organisms. And then you can look at the sequence which is conserved in any of those. And that's supposed to be shown here. If you look at this, actually the green box right here, you'll notice that there are lots of vertical red lines. Those are identical nucleotide sequences to each other between humans, Methanococcus, which is an archaea, E. coli and a bacteria, and then humans down here at the bottom. So here, conserved, that means it's something which is likely to be important for every single one of these, from a genetic point of view and certainly an evolutionary point of view. On the other hand, there's some regions right here in the red box which are not so similar relative to each other, and this is how you can start to build some of those trees, and this is going to tell you something about the differences between those different kinds of organisms. Difference in sequence, difference in organism, trying to find a correlation between those two. So what things are similar? And again, unfortunately, we're now looking at the crown groups of eukaryotes. Our textbook is very eukaryocentric, which makes sense. But again, we'll talk a lot more about it. Whenever you want me to slow me down and say, Dr. Stedman, what about the archaea? And I'll probably go on for like 10 minutes. But here, at least, we're talking about eukaryotic cells, and bacterial and archaeal cells are actually pretty similar here as well. If you just look at the top here, all of these cells should look very similar to each other. The bottom, these things should look really quite different from each other. All these things start as cells, and bacteria and archaea are also cells. Viruses are way cool and a totally different story. But all of the cellular life basically starts out looking pretty normal and boring. And how you get to these much more interesting and attractive things down here at the bottom is how that individual cell will make more cells, and in each of those cells turn on different genes, turn off different genes. And that's what gives you this amazing diversity down here at the bottom. How do we know that it's all about the DNA? Couple of different ways. Of course, the viruses, this is the most important and interesting one. When most people talk about DNA, they say, well, how do you know that it's just DNA which causes an organism to form? They'll usually use this example. Who are these two sheep? Dolly. So if you just take the DNA from an egg cell, put it into an egg cell that is lacking DNA, you can actually end up with a cloned organism. There are some issues with cloning that, again, we can talk about later. It's just the nuclear DNA, it's not the mitochondrial DNA, etc. So that's one way. Originally, and we'll talk a little bit more about this later on when we talk about the discovery of DNA as the genetic material, it had to do with bacterial viruses injecting their DNA into cells and that DNA being able to form more viruses. Again, most people seem to think they knew about the central dogma. What are the problems with the central dogma? It's the main problem with the central dogma. Viruses, of course. DNA goes to RNA, goes to protein. Well, RNA can actually go to DNA, retroviruses. So that's the big issue that people have with the central dogma. But the basic idea here, and this is all about information. And people can think about, a lot of people talk about information in the digital age. This is information in the biological age. But in terms of thinking about information transfer, you've got DNA, which is all you need, right, because of Dolly and the viruses. DNA is the information that is used to make more DNA. DNA is the information which is used to make RNA. RNA is the information which gets used to make protein. So as I just mentioned, reverse transcriptions is RNA going to DNA. What about reverse translation? Anybody heard about reverse translation? There are actually various theories about why reverse translation can't work. And the main one has to do with the genetic code that we'll talk about in a couple of weeks. And there's a really nice article, I've listed it down here at the bottom, Eugene Koonin. Again, I don't expect you to know this, but if you're interested in looking into it in more detail, he talks about the impossibility of reverse translation because the protein code, how many amino acids are there? 20 normally coded amino acids. How many bases are there? 4. Although in general you can always say they could be 5 because uracil versus thymidine. But that process going from 4 to 20 actually loses information because the genetic code is degenerate. So you actually can't go back. Once you've gone to the amino acid language, you can't go back to the nucleotide language, at least not in an unambiguous way. Whereas you can for RNA going back in the opposite direction. So DNA goes to RNA, goes to protein. Why do you get some cells that form sea urchins, some that form mice, and some that form seaweed? Has to do with the environment and what those particular genes are that happen to be in each of those individual cells. So it's a combination of DNA, just that information which is there. Now, one cell, the fertilized egg, has all of the DNA information to make a whole organism, but a nose is different than a stomach. How is it different? That's the environment that that particular cell happens to be in, which determines which of these RNAs are made and which of those RNAs are being made into protein. So what about DNA? Again, we'll spend a lot more time about this, but a couple of really important things that bear repeating. DNA is polar. Now, all of you have had chemistry. What is polar? Polar means you've got separation of electrons, etc. That's not what we mean in molecular biology, so just forget that temporarily. I know it's hard. When I talk about polarity in this class, I'm talking about one end to another end of a molecule. And so if you think about DNA, RNA, protein, all of these are polar because they've got a front end and they've got a back end. And front end and back end for nucleotides is 5' and 3'. And I'm going to be talking about 5' and 3' a whole lot in this class. So if you don't get 5' and 3', come talk to me, look it up, etc. So 5' to 3'. That's your polarity. And often try and have like one arm that shows this. DNA in particular is interesting because it's got a front end and a back end of each molecule, but usually those go together with something with the opposite polarity, front end and back end, again 5' to 3' and 3' to 5'. So that's the symmetric nature of DNA. One goes this way and one goes that way. So we're very symmetric relative to each other. And hopefully all of you know why we end up with this symmetry. It's because of the complementarity. Certain base pairs are forming with each other. Got an A on one strand, you're going to have a T on the other strand. But always in the opposite orientation. You're not going to have sequences which are both parallel to each other. So it's always going to be that opposite orientation. And we'll look at that again in much more detail a little bit later on. So because of this complementarity and opposite sequences, it's actually pretty straightforward to understand how you get more DNA. How does replication take place? You've got one copy here, separates into two strands. That information can be copied. And it's nice because it's redundant information. If you know the sequence of one of these pieces of DNA, you know what the sequence is of the other one because of this complementarity. So you pull them apart, you can replicate them, you end up with two copies of that, actually really 4 copies of the 2 copies of the information that you started with in the beginning. This is called templated polymerization, the process which is shown right here. You have the individual nucleotide. This individual nucleotide here also has polarity to it. So the polarity of the individual piece that fits together, the individual monomer, then gets put together into a much longer piece, still has this same polarity. So polarity of an individual nucleotide, polarity of a whole strand, and each of these things get put together by the DNA polymerases that we're going to spend a lot of time talking about a little bit later on. So that's DNA replication. Next step in the central dogma is transcription. So DNA going to RNA. The actual process of transcription is basically identical to the process of replication at a chemical level. It's taking individual nucleotides and adding together in a string in a polar, in a molecular biology sense, mechanism based on, again, one of these templates. So 2 strands come apart. These get made copies of. The big difference between transcription and replication is that instead of going from 2 copies of the information to 4 copies of the information, now we've gone from 2 copies of the information to many, many copies of the information. So transcription is very different from replication, and it's an amplification step. So you start out with 1, you end up with many copies. And it's that process, 1 going to many, that is basically the subject of the last 3 weeks of the course. That whole regulatory aspect. Because that's what makes an archaean, a mouse, a human, is that differential amount. Sometimes you have a lot, sometimes you don't. So transcription, I talked about this already, it's a templated polymerization. The other difference everyone will say between RNA and DNA, you've got uracil instead of thymidine. We'll talk about why that is. It has to do with DNA repair later on. Although whenever I say why, you should always go, Stedman, you shouldn't be asking why questions in biology. The answer is it works. And the answer to any why question is evolution. Right? Okay, so we have a hypothesis for why we think uracil is present in RNA rather than in DNA. And we'll talk about that a little bit more. But one of those hypotheses actually has to do with repair and the repair mechanisms, what happens to DNA. You have ribose instead of deoxyribose. That's why it's R instead of D. And we'll talk about that a little bit later on as well. Everybody talks about RNA as being single-stranded. I hate that term. That's why I have it in quotes on my slide. Anytime you see a Stedman slide that has quotes on it, it means he doesn't believe it. Now, why? Why do I have a problem with single-stranded RNA? It's because almost all RNA looks more like this, up here in this upper right-hand corner. Almost always the RNA bases are paired with each other. And this is even true in messenger RNA, certainly true in transfer RNA, definitely true in ribosomal RNA. So they're paired with each other. And there actually really are 2 strands. I like to think of RNA much more as a single molecule rather than a two molecule. So again, as I mentioned for DNA, it's usually two different molecules, one 5' end, one 3' end, two different polarities that are coming together. Here, almost all RNAs, there are exceptions to this, but for the most part, this RNA is a single molecule. It's got one 5' end and one 3' end, but a lot of those base pairs are interacting with each other. So it's not really a single-stranded form. Partly because of that, what happens to RNA when you have a single molecule and you're causing it to base pair? What happens to that RNA? What's the next thing on my slide here? I know it's early in the morning, you haven't had all your coffee yet. Gets you structure. Structure means what? Function, yes, good. I've learned something here. I don't know, maybe it was high school. But structure means function. And that's one of the really amazing things about RNA that we're learning more and more about as time goes by and as research continues, is that RNA is actually very, very structured and has lots and lots of important, even catalytic functions that we thought were much more RNA-based to protein-based. A lot of enzymatic activity that people always thought were proteins turns out to be RNA. A lot of regulatory processes that people thought were all proteins turn out to be RNA as well. So lots of interesting things about RNA, and as I mentioned right at the beginning, we could easily spend a whole term talking about RNA. From RNA, central dogma, we go to protein. The process of making proteins, and again, talking about RNA as being catalytic molecules, the first place I think that was really convincing that people showed that RNA had catalytic activity was in looking at the ribosome. The ribosome is almost completely RNA, but it's critical to making all proteins and has nice catalytic activity. So translation in and of itself, yes, it's making amino acid chains, which are then going to be your proteins, but it's almost all RNA. So the RNA activity and that RNA base pairs to itself, base pairs to other RNA, make structures which are going to give you functions. How do you go from a information, which is in a nucleic acid information, to a amino acid? Of course, is the translation through the genetic code. Again, quote, universal. Most organisms use the same genetic code, but there are some really interesting exceptions to that, and we'll talk about some of those as well. Hopefully all of you know that it's 3 nucleotides per amino acid. If you don't, then again, we should probably talk a little bit later on. And it's the transfer RNAs and the ribosomal RNAs that do this. You look at the ribosome. Here's this ribosomal structure. Blue up here is the large subunit RNA. Green down here is a small subunit RNA. Here are the tRNAs which are in here. And one of the things that's happened, again, since the last publication of the textbook, we learned a lot more about the structure of the ribosome, very much at a high resolution, to understand exactly how translation is taking place and how it's being regulated. Proteins we're not going to talk about very much in this class, with a few exceptions, mostly having to do with how those proteins are actually regulating transcription, or how those proteins are now being used as enzymes to make more of the nucleic acids. But once you have this stretch of amino acids which has been made by your ribosome, it can come together again, form a particular structure which will have a function, and in many cases that's an enzymatic function which is formed here by basically lots of pieces of protein coming together to make a relatively small, chemically extremely defined part of your protein, which then serves as a catalyst or an enzyme, or for that matter, binding to DNA, etc., that we'll talk about much more as we move along here. So molecular biology, what are we going to be talking about again for the next, how many lectures? 29, minus the 3 exams, is really about this process whereby you go from the information which is present in individual nucleotides, the sequence information here that's present in your polynucleotides, and again, this is dependent on that polarity, 5' to 3', That's used to make more polynucleotides. This is what process? Two different processes that I just talked about. Transcription and replication. Okay, now this is kind of a feed in. What's this one? Nucleotide information going to amino acids to make proteins. Translation. So these are the information processes. Replication and transcription, translation. Then, what else are we going to talk about? We're going to talk about how this actually happens. We'll talk about the proteins that will make individual nucleotides into polynucleotides. We'll talk about the proteins that help with the amino acids going to proteins, and also which ones are regulating these kinds of things. So one way that I like to think about this whole process of molecular biology is, have you heard about concept maps, the whole idea of a concept map? Just thinking about where all the different pieces really helps with the vocabulary. Sometimes it ends up being alphabet soup in this class. So when he talks about TF2D, where does TF2D fit in here? Okay, TF2D is a protein that's important for gene regulation. So have a picture that has these kinds of things on it, and then fill in each of those individual pieces. And where does TF2D go? Does Pol2 go? Etc. So how all of these things actually end up fitting together. I'm going to finish up just talking a little bit about genomes. What's a genome? This is a wonderful slide from Jeff Singer. Anyone seen your genome? Exactly. So unfortunately, people don't always completely agree on what a genome is. When I'm going to talk about a genome here, I mean all of the genetic information that a particular organism has. A lot of people talk about a genome as being all the genes that are present in an organism. That misses out on all of the other things that are present in a particular organism. So when you talk about a particular genome here, like the E. coli genome, it's not just the, I think it's 4,300 protein coding genes which are present in the E. coli genome. It's also all of the other nucleic acid sequence which is there, the regulatory sequences, etc. So when I talk about a genome, it's going to be the complete nucleic acid content of any particular organism. So again, in the case of E. coli, we've got 4,639,221 nucleotides, base pairs thereof, both strands. I'm not going to ask you all those numbers. What I may ask you is how big is the genome of E. coli, and say 4 million. I think that's pretty reasonable. How big is the human genome, by the way? Guess? It's about 6.2 billion if you talk about the haploid form, diploid form, excuse me. Haploid is actually about 3.1, 3.2. People actually argue about this. And so again, when you have, here, look, it's the E. coli genome. It's, 4,632,321 base pairs. That's one particular strain of E. coli that's been sequenced at one particular time. So this is going to vary. And it varies quite a lot from organism to organism, even just from strain to strain. When we talk about DNA repair and DNA replication, what we'll see is that there's actually lots of changes that happen. Any time that you replicate through about 1 billion bases, you're going to make a change. So anytime you talk about a genome, or the genome of an organism, it's always going to be a little flexible in terms of the exact sequence which happens to be there. Why do I have an elephant down here in the corner?
Because all that is true for what colibacilli is true for the elephant.
Quote of?
It's Jacques Monod.
Right. And actually, it's not what he said. It's ce qui est vrai pour les colis, c'est aussi vrai pour l'éléphant. But yeah, the translation's pretty good. But yes, exactly right. No, the idea is if it's true for E. coli, it's true for the elephant as well, which of course he meant humans in that particular case. There, of course, it's not completely true. But when we think about it from a molecular biology point of view, and I'll tell you a great Jacob Monod story a little bit later on in the term, is that From a molecular biology point of view, certainly if you're talking about the DNA, you're talking about the RNA, you're talking about the protein, yes, it is exactly true for E. coli and for the elephant. E. coli has one really big advantage, just thinking about it from the genome point of view. What did we say? 6 billion, 4 million. It's a heck of a lot easier to understand 4 million than there is 6 billion. So that's the main reason that I have it here. Thank you for pointing that out. Okay, so let's see. We've got how many more minutes here? I'll just finish up with this here. 2 slides, and then we'll call it quits, and we'll start again here on Wednesday. So one interesting question that they spend quite a long time talking about in the textbook is how many genes do you need to make an organism? So again, getting back to this point about E. coli and the elephant and looking at a simple system, well, E. coli is actually a relatively large bacterial genome. How small can you go and still have an organism which is functional? And so this is, I think, as far as I can tell, the latest, smallest version, although people argue a little bit about this. In the book, they say fewer than 500 genes are required. And then 200 to 300 common genes that you find in all organisms. Carcinella rudi was discovered actually a little over 10 years ago now. It's a symbiont, so it lives inside another cell. In this case, it's an aphid cell, which has a little under 160,000 base pairs. So, actually 170,000, excuse me. If I could read my slides, that would help. A little under 170,000 base pairs. So you can actually have a functional cell with a little over 160,000, around 170,000 base pairs. Now, admittedly, this is a symbiont, so it's very, very dependent on the organism which it happens to be living in and getting a lot of stuff from. When you all take virology next term, we'll hear about some of the viral genomes, which are a lot bigger than this, which is really interesting. What's the difference between a virus and a cell? But here, there are a couple of things which seem to be very, very important in terms of, A, these are some of those common genes that we've talked about. Almost all of them are involved in amino acids in some way. Translation, that ribosome, all cells have ribosomes. All of these ribosomes are similar. Lots of these proteins are going to be very similar as well. That seems to be something you can't get rid of. Amino acids seem to be very important. Amino acid metabolism seems to be really important. If you just look at a plot, and this is from their paper, the red dot down here is Carcinella in terms of size of the genome. E. coli is over here. This is millions of base pairs. GC content is not really important. But there are microbes, this is bacteria, that have up to 10 million base pair long sequences. And one thing that I always like to remind people, there's probably 100 times more genes in the microbes in your gut than you have genes. And the very last thing to talk about today is, well, if you've got all these, you know how small you can go, maybe you can actually make a cell. Can you, and this is to quote or paraphrase Craig Ventnor, can you play God? Can you make life? And he's actually trying to do this with some of his companies. So they have JCVI, it's the J. Craig Ventnor Institute, SYN 3.0. This was published 2 years ago now. They started with mycoplasma, which is a relatively small bacterial genome, but it's non-symbiotic, can actually grow outside of other cells. It's only about 500,000 base pairs. And they were actually able to shrink it down. So now it's only got 473 genes and still is functional. The thing is, they don't know what most of those genes are, and they actually just took genes that were present in the mycobacterium and just basically shaved off all the ones that they could and saw what they got left. So they're actually down to under 500 genes in a at least semi-synthetic organism that they've put together here. So we'll talk a little bit more about genomics and actually where some of these genes come from on Wednesday. Again, more questions, come find me now after class or over in SRTC.